Hi guys, I am Sarang Anandrapawat. Today in this video we are looking the information about the central dogma in life. Then what is the central dogma in life? It is the theory of stating that the genetic information flows only in the one direction. That is the DNA to RNA, RNA to protein, or directly RNA directly into the protein. This is the main definition of the central dogma. It is just stating that the genetic information flows only in one direction, that is DNA to RNA, RNA to protein, or if there is the genetic material RNA, then directly into the protein. This is the main theory. After that, the what are the processes involved in the that central dogma processing? That is the transcription, translation. These are the main processes involved in the central dogma system. Then these are the we are looking the information about the translation and transcription in brief. First of all, we are looking about the transcription. Transcription means it is the process by which the mRNA mRNA synthesis by the DNA template. DNA is double-stranded molecule by which by the process of transcription, the DNA molecule is from the DNA molecule we can prepare the mRNA, and that mRNA when the mRNA is produced, in that process the various enzymes use, that is the for the opening of the double stranded helix of the DNA, that is the ligase enzyme is used. And the second process is the assembling of the set of enzymes and the gathering of the nucleotides. This is the main process. And the whenever the DNA double stranded DNA is converted into mRNA, the uh, instead of the thiamine, that is the in the DNA, adenosine, guanine, thymine, and cytosine is present. After the it gets converted into mRNA, then the adenine, guanine, thymos, uh, thymosin is replaced by the uracil. This is the main difference between the mRNA is transcribed into the DNA is transcribed into the mRNA. And mRNA is the single stranded molecule, while the DNA is the double stranded molecule. After the mRNA is produced in the nucleus, produced or synthesized or the translate into the mRNA, then the mRNA is transferred into the cytoplasm from the nucleus. Whenever it gets goes to the cytoplasm, there is the mRNA has the 5' end and 3' end. 5' is end is known as the methyl cap, or the methyl group is present on that 5' end, so it is called known as the methyl cap. And at the 3' end there is the several amino groups are present, that is the AAA, so it is known as the A sites. So after that, the 5' there is the methyl group, so there it is the main active site, and there the ribosomes which are present on the rough endoplasmic reticulum, these are attached to the 5' end. And whenever it is attached to the basically the structure of the ribosome is composed of the large unit and small unit. And whenever it gets attached to the 5' end or the methyl cap, then the translation processes start. And this is the smaller unit and this is the larger unit, and in between that the mRNA is present, and it gets translated. Translation means is simple, the different difficult to understand this converted into the simpler form. Then it gets hold in between the mRNA, between the large unit and small unit, and it gets translated, and it gives the proteins, or now amino chain of amino acids. It is known as proteins. And this is the simple protein. And after that, the proper configuration if the after that the proper configuration of the that amino acid is occurred, then it gives the functional protein. This is the all about the central dogma, or the process of the central dogma, how it is works. Thank you so much. We will meet soon.